best estimate, there's around 100 million sea stars out in Port Phillip Bay. So some of my PhD work will also shows that they eat basically anything they come across. So anything that's living on or just below the sediment, living on pier pylons, if they can get their arms to it, they're going to try and eat it.
Welcome to Ascend Your Story from the Ascent Media studios in the sunniest place on earth, Sunshine, in Melbourne's west, on the lands of the Wurundjeri people. I'm Sam O'Reilly, the director at Ascent Media. We love the ocean here, and that's why so much of our work is focused on telling its stories and the people who protect it. Our guest today is a postdoc at Deakin's EcoGenetics lab. He uses molecular tools to tackle biosecurity challenges, from studying what invasive predators eat to detecting species with eDNA and developing new ways to find and remove them. He also applies these methods to understand bigger ecosystem patterns like biodiversity and the genetics of native and invasive species. Dr. Morgan Ellis, welcome to Ascend Your Story.
Thank you for having me.
So Morgan, did Seinfeld's marine biologist episode ever come up in your work, and have you ever had to pull a golf ball out of a whale?
I'll be honest, I'm actually not a huge Seinfeld fan. I grew up watching more Frasier than I did Seinfeld, but it definitely gets brought up a lot when at work. And you mention to people you're a marine biologist, they go, oh, yeah, so you a fan of Seinfeld? You're like, no.
Cliches.
Yeah.
When people find out that you're a diver, do people bring up Octopus Teacher?
Occasionally, yeah.
Mm-hmm.
Obviously, it's like an amazing film, and it's great because it really got people interested in octopus and just how smart they are. Mm-hmm. And it's, a great sort of starting point to get people interested in other marine species too. You know, octopus are great, but have you heard about the cuttlefish down in Whyalla? And it could really help snowball the conversation and really like get people interested in marine species.
It came out soon after we got our dive tickets, and I'd mentioned to people that I'd dive. Oh, have you seen? And I'd say, The Octopus Teacher. I'm like, yeah. I'm like, yes, I saw it. It was great the first time, but the fact that you bring it up, everyone brings it up when I talk about diving. Yeah. So we got to know each other through AMSA, the Australian Marine Science Association conference this year, yep. And you were the chair for this year's conference. How did you get involved?
So I started in AMSA, it would have been about 5 years ago. I just joined the local state branch as a student member, and then sort of with the rest of the committee ran a few events here and there. And then my supervisor was the treasurer, and he stood down and said, do you want to be treasurer? And I was like, no, I just want to settle into the PhD. So I settled into my postdoc and just sort of take more of a back seat. And then Ali, who was the president at the time, went, do you want to be treasurer? And I was like, no, I'm just happy taking your back seat. And then we got to the AGM and nobody stuck their hand up for treasurer. And I went, yeah, all right, I'll do it. And then from there became sort of president of the Victorian branch. Mm-hmm. And the conference moves around sort of each branch, and it sort of came up to Victoria's turn, and me and Ali sort of started driving it and looked around and went, well, I guess we'll be the ones in charge of organising it. So, yeah, that's sort of how I ended up chairing the conference.
It was a big conference, like so many rooms, 5 rooms.
Yeah, I think on Monday we had 5 rooms and about 600 delegates all up. And about 400 of those were presenting either orals or posters. So, yeah, I was really impressed with how it all turned out.
Well, we really appreciated you having us on board to do the photography, and we had a booth there, and it was great to connect with people from all around the country. I hadn't thought about the fact that it was a national conference, and I'm thinking, I'm just going to be talking to people about the bay and diving here. And it's like, oh, you're from Queensland? Oh, from WA. It's like, okay, cool. We're from all over. Now, the conference theme was harmony in marine systems. How do you think that shaped the conversations?
I think it put a bit of a sort of positive spin. A lot of conferences tend to focus on all of the issues in the environment, particularly the marine environment. There's obviously, with climate change and everything that's going on, there are a lot of issues. So they can sometimes get a bit of a negative spin. And I think sort of bringing it back to the idea of that, like, we're just trying to achieve harmony, whether it's sort of merging Indigenous knowledge systems with Western knowledge systems, sort of aligning policymakers with science, just to sort of bring that positive spin back to conferences and say, like, yeah, there are challenges in the environment, but all of these people are here because we're doing good things and we're trying to improve it. So let's focus on the improvements that we can make while acknowledging that there's challenges there, rather than focusing on the challenges and acknowledging that we're sort of making some improvements.
There was a real Indigenous element to the conference. Did it spur new conversations or collaborations?
Yeah, AMPS has been, I reckon, of all of the conferences I've gone to, one of the leaders in sort of the Indigenous marine space. We've always had a really good Indigenous working group that form a large part of the conference. And it's, really, it gives them space to drive the conversation where they want to go. And it was great having a plenary by Jodie Edwards, who was able to use sort of that Indigenous storytelling as a way of bringing us along and sort of emphasising the importance of talking with Indigenous and traditional owners as you move forward, and like thinking about that with your research, because there's just so much knowledge held in and amongst these communities that you're just missing out on otherwise. So I thought Jodie's plenary was really, really inspiring in that regard.
What was your biggest takeaway from the conference for yourself?
For myself, I think it was just that, you can actually put a conference together as a reasonably new ECR. But I think, overall...
Could you explain what an ECR is?
Oh, sorry, an ECR is an early career researcher. So I'm only about 3 years out of my PhD. A lot of the conference organisers that I've been to in the past, they're sort of more middle to late career stage. So, it sort of felt a little bit out of my depth at times, but there was a great team sort of supporting me. And I think that was the biggest takeaway for me personally, was that these sorts of things are achievable. Yeah.
Mm. Yeah, it was a lot of fun, and so many social nights. I was very disappointed to miss out on the karaoke night.
That was so much fun.
There's something you need to know about me. I love karaoke. Now, the day after the conference, there was a field trip to go snorkeling in the bay here, which I was lucky enough to get out on and photograph the seals and the weedy sea dragons and the delegates that got out there. Now, next year, the conference is in Canberra. What kind of field trip do you think they'll be running then? Do you think it'll be a swim in Lake Burley Griffin?
Yeah, I've suggested snorkeling out in Lake Burley Griffin. I don't think it was taken up too rapidly.
Yeah.
Maybe go out paddleboarding or something.
That sounds like a good one. Now, what got you into marine science?
When I was probably... It's a typical story when you're like 4 or 5, wanted to be a marine biologist. And I just completely went off the idea, wanted to do psychology for a little bit. And then when I was in year 11 and 12, I really wanted to get into environmental law and started sort of working towards it. And then someone tapped me on the shoulder and went, "Why do you want to do law? Nothing about you is law-based. Like, why do you want to sit behind a desk?" And I went, actually... Yeah, you're right. I'm much more interested in science. So I was flicking through one of the uni catalogues, and Deakin had a marine science course, and I went, actually, that sounds like a lot of fun. And I'd sort of just started getting into diving, so I was, getting a better appreciation for the marine environment, and it sort of just, clicked that this is the way I wanted to go.
Where has your research taken you in the field?
Actually, quite a lot of places. So locally, we sort of work quite closely with governments and stuff all around Australia, but I've done sort of field work across most of Victoria. I've also been lucky enough to head over to Perth to do some field work out on one of the Navy bases over there. And we've recently started working with some of the Indigenous biosecurity ranger groups up north. So I've been up to the Torres Strait, went out to Cape York with some of the rangers there, which was a crazy experience. We've also done a little bit of work with a UNESCO-led project out at Fiji, which is all about sort of trying to build biosecurity capacity in Fiji and sort of get that local capacity to detect invasive species rather than sort of relying on samples being sent, back to Australia or to other places, which just slows things down and makes sort of being able to get on top of invasive species a bit harder.
Have you been involved with CSIRO at all, with the ship?
No, I haven't. There's a PhD student in our lab had been out on the Capstan project, and I was very jealous. Yeah, I sort of missed a lot of that during my PhD because of COVID.
Got to chat to them through the conference, and would love to get on the ship, take some photos. If you weren't a marine biologist, what job would you want?
Oh, that's a hard one. I reckon.
I'm sure you'd love to go back to law.
Yeah, definitely. Sitting behind the desks was a favorite part of science. I reckon I would run a little cafe somewhere, just like make good coffee, good cheap meals, nothing too sort of big and fancy, just sort of that nice home style hearty meals in a little cafe somewhere quiet.
I don't know if it's like this now, but I felt like Geelong needed a good cafe a few years ago.
Yeah. Trying to find a good coffee is not as easy as it is in Melbourne.
Yeah, maybe like on a Sunday.
Yeah.
It'd be nice. What's your favourite marine species?
Look, it changes every time I'm asked it, but it mostly between either the weedy sea dragons or the cuttlefish. I think they're just, both of them, they're just so colourful and so like crazy looking. They're almost like alien looking type species swimming around. And I was lucky enough to swim with the cuttlefish a couple of years ago, and just the way they can change the colours and the texture of their skin and at the big aggregations in Whyalla, just how many there are swimming around, and that they just don't care that there's a diver swimming around pointing a camera in their face. And it was, that was pretty wild, pretty mind-blowing.
Yeah, I was out there just a few months ago, and they're just so chill.
Yeah.
Occasionally they'd pop their heads up and go, what are you taking photos of me? I'm in the middle of lovemaking here.
Yeah,
then they get back into it.
Try and ignore that part when you're taking the photos and be like, I'll just pretend that that's not what they're doing.
Are they fighting or... yeah. So you've done a lot of diving in Australia, I assume?
Yep.
What's your favourite diving spot in Australia?
I reckon my favourite, and it was, luckily enough, it was my first open water dive, which I think probably contributes to it being my favourite. It's Blair Gowrie Pier, just down on the Mornington Peninsula. There's so much life under there. I've seen cuttlefish, I've seen sea dragons, seahorses, and there's about 50 different species of nudibranch. So I've just got like a little TG6, which is, a great camera for macro photos. So I just love getting down there and, spending an hour and making it probably 20 meters down the pier, if that, while your buddy swims off, does the whole pier, swims back, and you go, all right, must be time to head in, just because you're sitting there taking so many photos of all of his nudibranchs. It's by far my favourite site.
And amazing during the day, but even more spectacular at night.
Yeah, when all the dumpling squid come out, and it's amazing how much colour there is down there.
They're just the cutest things. I've only seen them once, and just the way they pop their little arms out, looking like they're having a fist fight. And what's your favourite diving spot outside of Australia?
My favourite one so far has probably been when I was in Fiji. So I flew in after my supervisor, and he sort of messaged me and went, "Oh, I'm going out diving tomorrow if you want to come along." And I had about half an hour's sleep on the flight. I was like, "Yep, no worries." So I went down to this little local bus stop, ended up in sort of the middle of nowhere, no phone, no idea where I was, managed to find him in the taxi, went down there. And we went out to this place called Shark Reef, and you could see all the sort of shadows swimming around, jumped in, and there was a 3-meter bull shark just chilling in the water. And again, didn't care that we were there, sort of swam around, saw a whole range of sharks. And we're doing our safety stop, and you're sort of just below the reef flap. So you're sort of peering up, looking at all the reef sharks. My supervisor tapped me on the shoulder and told me to turn around, and the bull shark was back chasing fish, and like getting to see them hunt out in the wild was cool. And then the guide tapped us on the shoulder and went, "All right, back to the boat." And we looked at where the boat was and looked at where the bull shark had been hunting and went, "Oh, we have to swim over that." It was a tiniest little group of divers swimming back to the boat.
When we're in South Australia for the whale, when we went to South Australia to dive with the cuttlefish, we went over to the other side of Adelaide to find the leafy sea dragon. And when we were swimming back to the pier, I'm just swimming across the surface, and when I get there and I look, and the guide is he's under the whole time, and I'm like, why did you go under the whole time? He goes, didn't want to meet the locals. I'm like, and it took me a second to realize what he talked about. And I'm like, yeah, you don't want to be swimming on the surface in South Australia.
Yeah.
You don't want to be swimming over the top of sharks. I've only dived with sharks once, and it was at Melbourne Aquarium, where you don't really get to dive properly. Like there's no, you don't get fins. They don't give you any controls. All they teach you is how to clear your mask and clear your egg. But the training, they've got a little platform near the surface, and you stand on that, and they test you on how you're clearing things. And while they were testing somebody else, they said, "Oh, you can put your head down and have a look." So I put my head down, and this little reef shark just starts heading towards me, and at the last second, just and I was freaking out a little bit, but I'm like, it's okay. It's a controlled environment. Just think of it like it's a movie.
Just testing the tourists out.
A couple of minutes later, the guide comes over and she's like, she puts her head down as well. Shark comes back, comes straight towards us. Last second, turns away, tail whips me. Anyway, we come to the surface and I'm like, wow, that was pretty full on. And the guide was like, I know, I was shitting myself. I'm like, hang on, what? That's not a controlled environment when the guide's a bit worried. So yeah, but yeah, we're planning a trip to Southwest Rocks in November, and I've heard of schools of hammerheads, which just sounds amazing. I'm gonna shit myself, I'm sure, but at least I'll be able to tell my kids to swim with sharks properly. Yeah. How do you explain your research to people outside of science?
So a lot of people with sort of Australia know a lot about invasive species. There's obviously a strong sort of biosecurity interest at the airport. You have to declare all of this sort of stuff. But a lot of people don't sort of realise that the marine environment's an equal problem. So it's sort of handy having that as a sort of background to be like, all right, you know about rabbits and cane toads. I work on those sorts of species, but in the marine space. And funnily enough, COVID really helped start to explain things. So one of the things we can do to detect invasive species is use eDNA or environmental DNA. And we collect the water sample, filter all of the DNA out from it, and then basically run the same thing as a COVID PCR test. But instead of sticking a swab up your nose, we have the DNA stuck to this filter. And then instead of looking for COVID DNA, we look for an invasive species. And we can pick up really small concentrations. And the advantage of that is then we can get the invasive species when they're in a small population before they sort of have that chance to establish. And it just makes trying to get people in to clear them out and eradicate them before they can get fully established a lot easier.
When we were at the conference, we saw CSIRO had a fancy machine that helped with testing for eDNA on mass. Is that the kind of machinery you use?
We use a lot simpler. So during my PhD, it was a syringe with a filter attached to it, and the filter's about like 0.2 microns or something. So you get sore hands sort of constantly pushing the water through. And now we have sort of small pumps, but we can do like one filter at a time, whereas the kit that the CSIRO have can just sort of be constantly monitoring. But it's the same principle at the end of the day, that we can use it to look at either a range of biodiversity with sort of a universal system and just see what's in the water, or really target it to sort of invasive species.
We've seen enough police shows to know that there have been a lot of breakthroughs in the usage of DNA in forensics. How has eDNA testing, has that progressed as well?
Yeah. In probably the last 5-10 years, it's gone from this sort of, I suppose you could do this, to now almost every, say, state in Australia has a molecular biosecurity water testing system. Sometimes it's only at maybe 1 or 2 ports, sometimes it's broader. And we've been working with sort of a federal government department, DAF, to really get some more remote techniques. The advantage of it is it's really simple. You can give it to sort of untrained citizen science type systems. you can, give people these filters and they can go and collect samples and send it back. So we're trying to get sort of more remote biosecurity surveillance up north, where there's a lot of vessels that come in ports of first entry up north that sort of we don't have eyes on. So working with some of the local ranger groups, they can do all of this remote biosecurity surveillance. We've removed the cold chain. They can send it down to Melbourne, all just regular sort of shipping. And we can then sort of test it for invasive species there and sort of really help improve that sort of grey areas where we knew boats were coming in, but we just didn't have enough people out there to have eyes on what's happening.
Do you use any citizen science projects to get water samples?
I don't with my work, but there's definitely lots out there. We work quite close with Melbourne Water, and they had one a couple of years ago trying to find platypus. So they were working with EnviroDNA and got hundreds, if not thousands, of water samples from just citizens passing water through a little filter and mailing it back. And they were able to find rivers that they didn't realize there were platypus and sort of really get a really good distribution of what platypus were doing around Australia, which I think, it's such a good use of all of these people who are really engaged in science. And it's so easy to do, just running water through a filter. So I think it's definitely an area that we'll see more and more use.
So it sounds like you've got so much information in the tiniest amount of water. Like how much are you finding out from a droplet?
So with the species-specific approach, we can get down to 1 copy of DNA per microliter able to detect. But then we can also do this approach called metabarcoding. And so, all species have different DNA. We can amplify all of that DNA and then sequence everything that's in there. And you get something like 1 million sequences back, run it through a big computer system that will find all of the unique ones. And often we get sort of 100 different taxa coming up. So a lot of those we can't quite ID to species level because we need a reference database to compare that DNA back to. And a lot of the Australian species we just haven't sequenced yet. So we can get a lot of it down to sort of like a family level ID. But you get all of these taxa coming back and a really good idea as to the biodiversity swimming around sort of either rivers, the ocean, wherever you take a water sample.
Mm-hmm. How reliable is eDNA compared to physically observing species?
It will never be as good as physically observing a species. And we're in this sort of awkward space at the moment that we are improving with our molecular tools and our certainty around the molecular tools, but we haven't quite improved enough to say, all right, I've definitely got this invasive species because I've got a positive detection. Or you can sort of really know is I've got the DNA from this species coming up in this sample. We have good controls for contamination, but there's always a chance of contamination. But where it's really good is sort of triaging sites. So if you are worried an invasive species is spread and you've got 5 ports, you can, instead of trying to get divers into those 5 ports, taking a lot of time, do an eDNA run and go, all right, these 2 ports have come up with a positive hit. Let's focus on these ports. Let's get divers in. Let's really check these ports, and we'll put the ports where we had all of the negative detections back down on the list. And it really helps sort of triage that way. But I don't think we're quite at the stage where we can say 100%, yes, it's definitely here because we have an eDNA hit. There's just too many variables, sort of eDNA floating in the water. Is it something growing on a boat that's gone past, but it's not actually staying in the harbour? So, we're not quite there, but we're getting closer.
So when we leave DNA somewhere, it might be a hair or saliva or bodily fluid. How are marine species shedding DNA?
Well, more or less the same way. So animals are constantly sort of losing cells. Those cells are just floating in the water containing DNA, feces and mucus. We also get sort of free DNA where a cell's broken down and the DNA is just floating in the water. Sometimes it'll attach to like sediment or something. It degrades a lot quicker. So you can pick it up, but you sort of have to get there right as it's sort of breaking down. But almost sort of every animal's just losing cells, losing DNA into water basically the same way we are. And we're just lucky that it sort of floats a bit more in the environment. It's a lot easier to catch than trying to, sweep up a bunch of skin cells or a bunch of hair.
Is it detectable at any level, or does it fall to the bottom mostly? Or
so there's a bit of research sort of looking into that. We so like with the Northern Pacific sea stars, which is a big species out in Port Phillip Bay, they live on the bottom. We do detect it all throughout the water column. And when sort of not quite sure how it floats, how it sinks, it depends sort of what particles they might be binding to. But it definitely sort of does mix. But the closer you are to the actual organism, the better chances you're going to get. So, yeah, if you're, targeting a animal that lives on the bottom of the seafloor, taking the samples at the bottom of the seafloor, you probably get better results. Speaking
of Northern Pacific sea stars, when did we start calling starfish sea stars?
I've sort of always grown up calling them sea stars.
Okay. That's just me.
I think it's the same with jellyfish. There's a lot of sort of push to sea jellies, but then, seahorses aren't horses. So I'm happy to accept either sea star or starfish.
Because they're not actually fish. Sure.
Yeah.
So Northern Pacific sea star, why are they so harmful?
So they're listed by the IUCN as being one of the 100 world's worst invasive species. A single female can release 20 million eggs during a breeding season. So you don't need a huge population and you don't need a very long time before you've got a huge population swimming around out there. Some of my PhD work will also shows that they eat basically anything they come across. So anything that's living on or just below the sediment, living on pier pylons, if they can get their arms to it, they're going to try and eat it. So they form these really big aggregations, these really dense mats of sea stars, and they'll just sort of consume everything in their path. So they can be, really damaging in a really short space of time.
I joined a group to collect some at Port Melbourne, and I thought we'd picked up the biggest haul of all time, and they said no, that was nothing. I think we collected like 600 or something. They said no, we've collected 30,000 in the matter of 2 hours. I'm like, what? And they said they were just piled, a meter high. I couldn't imagine that. Like we were searching for them, so it must have been not the peak time for them, but they're pretty horrific, but so easy to pick up.
Yeah. But best estimate, there's around 100 million sea stars out in Port Phillip Bay. So they're easy to pick up when they're in close to shore, but there's a lot sort of in the deeper parts of the bay that obviously it's a lot harder to get people out to collect. And 100 million, that's a lot of people bringing in a lot of sea stars to try and eradicate them.
And if they're able to shed, say, like 20
million eggs. Yeah.
Wow. Okay. Yeah, that's an exponential growth right there. Wow. Now, what do pheromones and cane toads have to do with these sea stars?
So pheromones, they're these sort of small molecular compounds. Every animal uses them in some regard, even we use them. And the best way I can describe them is they let animals sort of sniff each other out. So often you don't necessarily know you're smelling somebody else's pheromones, but they sort of allow animals to communicate and find each other. And one of the big areas that they're used in is for sexual reproduction. So we have sex pheromones. And the Northern Pacific sea stars, when they breed, they form these huge aggregations. They all get in close because they're broadcast spawners. So they want to be in closer when they release all the eggs and sperm. It's all sort of in the same area. You're not releasing them at other ends of the bay. So we sort of went, well, we know about these pheromone traps that are used a lot in terrestrial environments. They're used in agriculture. So there's been some work around cane toads and stuff with them. But no one's done it in the marine environment before. So we thought these are an excellent species just as a proof of concept. Can we get this to work? They're easy to find in the bay. We know that they must be using some form of communication to form these aggregations. So we've started working towards trying to make the first marine pheromone trap sort of focusing on marine species.
And what does the trap look like?
We're not quite at that stage yet. Hopefully in the next 12 months, we'll have sort of everything designed and nutted out. But the designs that I have seen look sort of similar to a cray trap where the sea stars fall in and just climb back out. And then we'll have something sort of floating above it, just slowly leaching out these compounds that we've found that attract them. So hopefully we'll get sort of a nice big plume of pheromone and see all of these sea stars coming in. And then I think it'll sort of work as a positive feedback cycle. The more sea stars are there, the more they'll be releasing pheromones, the more sea stars you'll have moving in.
Any other animals attracted to it?
Not that we've found. So we've tested it on one of our native sea stars, the 11-arm sea star, which is probably the most closely related in the bay. And basically to test it, we put them in a chamber and they can either go towards the pheromone up one side or they can go towards the seawater blank up the other side. And if it's sort of no evidence of attraction, it's right on that 50-50, which way they choose, no different to flipping a coin. The Northern Pacifics, we see like 90% of them heading towards the pheromone. The native sea stars was almost exactly 50-50. They just, did not care that this stuff was being added to the water.
So the 11-arm sea star, not an issue for the environment?
No, yeah. So they're our native ones. If anything, they're real positive to have in the environment because they do seem to eat the invasive sea star. Not enough to sort of be a big biocontrol and eradicate them from the bay. But yeah, so they're sort of the bigger, spikier ones. A lot of people think they're the invasive ones because they're a bit scarier, a bit less attractive, but they're a really good species to have around.
So the Northern Pacific 5-arm?
Yeah.
And is that the biggest tell between the two?
Yeah, so that's the biggest between the two. But we have another native species, the granulated sea star. And when the Northern Pacifics first arrived down in Tassie, they actually thought it was the native one, so they ignored it for about 10 years. And in that time, it spread up to Port Phillip Bay. And the biggest way to tell the two apart, once you see them together, it's super obvious. But when you don't, it's a bit confusing. Northern Pacific sea stars have a really pointed tip on their arms, whereas the native sea star has a much rounded tip. And the invasive sea star also likes to sort of sit with its tips pointing up towards the sky, whereas the natives like to sort of be stuck firmly down onto the rock. Yeah.
Okay. And how did they make it to Australia?
They spread originally down to Tassie from sort of a Japan, Korea, China sort of area as larvae in ballast water. So just, little sort of baby sea stars came down, the ballast water got released down in Tassie, and as I said, they were misidentified, so ignored. How they got up to Port Phillip Bay is a little bit more sort of in argument. I think it was again by ballast water. There's some evidence that there was actually maybe a few individuals sort of living somewhere on the boat that then managed to sort of get free and start breeding. And 20 million eggs doesn't take many for it to be a problem. So it could have been a handful of individuals, or it could have been ballast water.
And if the general diver or snorkeler wanted to help with collecting them and eradicating them, do they need a permit or a fishing permit to do that, or are they allowed to just pluck them?
You often do need permits. So I think the best way, if you do want to help out, is get in touch with some of the local community groups, the sort of coast care groups. They often have collections. So they'll help train people, make sure you are picking up the right species, and they can help sort of point you in any direction, depending on what permits you need, if it's just a regular fishing license. And if somebody is out fishing and they catch them, the best thing they can do is sort of not cut them up and throw them back, which used to be the old advice, because they can then just grow from being cut up. But to bag them up in a plastic bag and put them in the bin is sort of the best advice if you end up catching them.
And washing your gear if you're diving with them. Yeah.
So following the clean, check, dry method, which works for fishing gear, diving gear, boats, you want to clean it with fresh water, ideally warm water. Obviously, a boat's a bit hard to clean in warm water. They don't really fit in the bathtub. Dry it thoroughly, especially areas that can sort of retain a bit of moisture, and make sure you're not sort of moving wet gear between areas that don't have sort of some of the invasive species.
Can you describe the main focus of your PhD work?
So one aspect was the pheromone trap. And then I suppose the other aspect is sort of building more tools to help sort of detect invasive species, which is where that sort of eDNA side of things come in. So we've been working with sort of different institutions and different governments to identify key invasive species, and then trying to create that eDNA assay to be able to find them. Because you don't want your assay to be detecting the native species as well. So to sort of...
You're going to have to explain assay.
Oh, sorry. An eDNA assay is where the DNA sort of binds when you're running it in the qPCR, and it's what highlights that the invasive species is there, basically. So you want that to be really specific to the species that you're looking for, because if it starts to sort of identify that there's native species there, you then get those false positives, and you sort of lose a lot of faith in the sort of that all of these tools are working as they should be. So for one example, like we ended up sequencing about 300 different samples from around Australia just to find about like a 30 base pair, which is like those basic units of DNA, a 30 base pair region that was specific only to the one that we were interested in and wasn't going to sort of cross-amplify anything else out there. So, yeah, there's a lot of work in there. And then just trying to sort of build that biosecurity capacity around Australia and working with governments to sort of streamline things, optimize the methods that they can use, the sampling protocols, and just sort of build a more robust biosecurity system that we can get more samples done for a cheaper price.
Mm. And why did you choose that focus?
So we learned about invasive species back when I was in sort of second year. One of the key ones that we learned about was the Northern Pacific sea stars. And then I was out for a dive in Port Phillip Bay with Dad and just saw this giant mass of them. And it was sort of like, well, we know they're a problem, but nothing's really being done about it. So that's, where my interest sort of started. And I went to my honours supervisor and went, I'm interested in these things. Can we put a project together? And that led to a PhD sort of on the same topic, looking at what they were eating and sort of some of the ways they were interacting with prey. And then, sort of from there to a postdoc in sort of a broader biosecurity and invasive species sense.
So you're looking at North Pacific sea stars. What other species are you looking?
So there's an invasive sea squirt, which...
I do not know what a sea squirt is.
They're a really hard one to describe. They're kind of like a sponge, but they're also really the opposite of a sponge. They're more closely related to us than they are almost sort of any of the other marine invertebrate species.
Wow.
But they just sit there and look like a sponge. They pump water in, they pump water out. So we've been looking at one of those, which has recently been detected in Australia. We've also done quite a lot on the Asian shore crabs, which we first detected in Port Phillip Bay around 2021, and then realised they'd actually spread back in 2018 from things like photos up on iNaturalist. We were able to sort of re-establish that point. And then we, sort of looked at some of their diets and eDNA surveys to see where around Port Phillip Bay and outside of Port Phillip Bay they may have spread. There's an invasive seaweed, wakame. We've done a bit of sort of similar work on that. So, there's no shortage of invasive species. There's about 180 introduced ones in Port Phillip Bay. So there's, plenty of work ahead of me, I'm sure.
So once you've done all this research, who do you pass that information on to?
So we largely work with sort of government and management authorities. So if we detect an invasive species where we haven't detected them before, we'll talk to the state government, places like Parks Victoria, so that they're aware of it. They can get divers in and either confirm it or try and eradicate it. If it's one that hasn't been in Australia before, then we can notify the federal government as well. And then, it's sort of just the interested parties that we're working with. So maybe it's a port that we're working with, or different state governments, different government departments. But yeah, sort of if we find them, it's up to the management authorities to decide sort of where to take it from there.
So what is the central question you're exploring in your postdoc?
I suppose if you had to sum it all up, it's how we can better detect and then do something about invasive species. So it's one thing to know they're there. It's another thing to actually do something about it, which is why this pheromone trap's so important. Is a lot of species, we're not even at the Stone Age when it comes to eradication. It's all, we're still using our hands to remove sea stars from Port Phillip Bay. Some of the other species, we have slightly better methods. You can wrap plastic around pier pylons, but then you kill everything else living under the pier pylon. So we're so far behind the terrestrial environment when it comes to these sorts of things. So yeah, I suppose the big interest is how we can start to get some eradication tools and sort of better manage and hopefully start to eradicate invasive marine species.
So robots?
Yeah, that's one method. So up in the Great Barrier Reef, and it's not an invasive species, but it's a pest species, cots. They've tried using a robot that can use AI to identify it and then stab it with vinegar, and basically the sea star just dissolves away. They found it works.
Vinegar is good for everything, isn't it?
The robot works really well, but it's quite expensive compared to a volunteer diver who's just going to swim around and do it anyway. So yeah, I think definitely as we move towards cheaper robotics and cheaper and sort of more reliable AI, I think that's going to be a huge market that opens up for controlling invasive species.
I'm just picturing one of those creepy crawly pool cleaners just going around. What do you hope policymakers or industry leaders will take away from your findings?
I hope it just starts to, for starters, raise awareness that this is a big issue. As I mean, like a lot of people see the ocean as being out of sight, out of mind, when invasive species have been spread around the world basically since we've been getting in boats. So it's a huge problem and it's not going away anytime soon. So I think realizing that it's a problem and that going into the future with uncertainties around the environment, around climate change, it's more likely to impact fisheries. It's more likely to impact tourism. If the wrong species arrives in the wrong space, it could wipe out, entire industries. So sort of getting that awareness out there that this is a problem and that we need to start better sort of managing our marine environment, better sort of stopping these species from establishing in the first place, and being able to do something when they do establish is, I suppose, the main key sort of message for policymakers.
Have you had any big wins?
Yeah, so I guess like with the pheromone trap, when we started out, it was a sort of year-long project with the idea that hopefully it would snowball. But right from the start, we went, look, we'll see what we can do. We're going to take a water sample. We're going to see what chemicals are in that water sample, and we'll see if one of those works. I don't think any of us actually expected to find compounds that were going to work with 90% of the sea stars. So that was, a huge win, seeing that this method is actually working, that we're relatively, I think, a lot easier than the research group were anticipating, managed to find these compounds that started attracting the sea stars. So that was, a huge win sort of early on. And it's just allowed the project to sort of keep going to the point that we're almost ready to sort of start getting traps in the water.
Yeah, great. So how long until you think you'll have traps in the water?
We're aiming for the next breeding season. So they breed sort of over the cooler winter months. So hopefully sort of June, July next year, we'll have traps in the water as sort of a first field test. And then if they work, we can hopefully start sort of rolling them out to governments, to community groups that are interested reasonably shortly after that.
Great. Look forward to going for a dive and seeing big traps. Tell me about the seagrass restoration out in Western Port.
Yeah, so like the other part of our lab, so I sit on the biosecurity side. The other part of our lab do a lot of restoration work. So marine environments are hugely degraded. We've lost something like 80% of seagrass from around the world. So we're similar to biosecurity. We don't have as good tools in the terrestrial environment when it comes to restoration, like the marine environment. So the lab has been trialing methods to better restore areas. And I'm doing a little bit of work with on the student looking at sort of the connectivity and the population genetics of the seagrass out in Western Port Bay. So seeing if maybe some sites are more adapted to change, more adapted to temperature, seeing if we can find sites that sort of might have a lot of clones of seagrass, so it might sort of lack that genetic diversity that's hugely important for disease resistance, and sort of seeing maybe if a population's putting out a whole bunch of seeds, like it's the source population, we need to protect it. So we're doing sort of quite a wide range of work out in Western Port Bay, trying to restore these, what used to be huge seagrass meadows and are now sort of very dwindling meadows in some pretty tough areas.
Mm. How's that affecting the weedies?
I imagine sort of hugely. Weedy sea dragons love hiding in seagrass. It's one of the easiest places to find them. And there's a pier over in Western Port, Flinders Pier, which is a pretty good spot you can go find them. So I wouldn't have been surprised if once upon a time they were found sort of pretty widely spread throughout Western Port. And then, with the seagrass loss and sort of the water quality and turbidity issues, I imagine that population would have sort of shrunk right back. So, maybe one day we'll restore all the seagrass and there'll be weedy sea dragons all throughout the bay.
With the field trip after AMSA, when we went over to Portsea Pier and saw all the weedies there, I was amazed with how many were there. I thought I was seeing a lot at Flinders, but seeing them all in the seagrass there was pretty spectacular. Unfortunately, we were snorkeling, so I'm not a free diver. Plus, I was in a 7mm wetsuit, so I was just bobbing on the surface. And just so many down there. It was great. Let's talk science communication. What's the biggest challenge in making marine science engaging for the public?
I think the biggest problem we've sort of touched on before is that the ocean's very out of sight, out of mind to a lot of people. They see the top of the water, there's not a lot there, and that's all they see the ocean as being. They don't understand how much life there is just below the ocean, how complex it is. I was diving with someone recently and they mentioned they were from Melbourne, and I went, we'll go for a dive. And they went, I didn't realize there was anywhere to dive around Melbourne. And you're like, we've got so much life and so much diversity sort of just below that top layer that I think that's the hardest part, is sort of getting people to realize how much more complex the ocean is than it sort of initially appears.
I've got friends that live in Rye who are divers, and I travel down to Rye to go, a good day trip. And sometimes go down there for a holiday to go diving. But these friends refuse because they're like, no, it's too cold. I'm like, there's so much to see. So much to see at your back door. Yeah. Do you think visuals like photos and videos are more effective than written reports for marine science?
Yeah, 100%. Because it brings all of that life and brings it to the surface and shows people this sort of alien world that's sort of, as you said, just right on the doorstep. And I think I've noticed this because I do like a little bit of photography on the side, just nothing like what you guys do, just sort of mucking around with a little camera. And you show people the photos and they're like, wow, this is amazing. Whereas if you show people like a photo of a landscape of a mountain, they're like, oh yeah, whatever, it's a photo. And I think it's because it's just so unknown, so unworldly. They've never seen anything like it before, that even sort of simple images are mind-blowing to them. So getting sort of good quality images, video of sort of, the cuttlefish as they're changing colours, sea dragons as they're swimming around, it really brings this other world up to people's sort of consciousness, and sort of they go, all right, maybe we need to do something about this. Yeah,
when I first started diving, I was blown away, I think. Yeah, getting cornered by a bull ray or smooth ray under a pier. And I was like, wow, this is a monster. And then sharing the photos on socials to my friends who had no idea. I had no idea before I was diving. And it's changed everyone's attitudes. It's an amazing place amazing asset that we've got in the bay.
Yeah.
Seeing blue-ringed octopus to the smooth rays to seahorses and weedy sea dragons. Like people travel all around, from all around the world to see the sea dragons at Flinders Pier.
Yeah,
I've met many international traveller there. It's like, I've been so excited to come here. And then you have to travel a little bit further to see the leafy sea dragon. But I was lucky to see one over there a few months ago in South Australia.
Yeah, it's not all coral reefs. There's a lot more life once you get outside of the coral reefs.
Yeah. Have you ever had to simplify your work so much that it felt inaccurate?
At times, yeah. I try not to. And I think as a lot of the time in science, people underestimate what the general public know and try to simplify things too much. Through sort of one of my colleagues' PhDs, we ended up at a primary school teaching them about DNA and eDNA. And these were, I think, grade 4 kids or something, and they just were straight on it. They didn't need sort of to see the DNA to understand all of these complex topics, and they understood all about the currents running around Australia, the Lewin current under sort of southern Australia, the East Australian current. And they
thank you, Finding Nemo. You've spread the word already.
Yeah, they had a better idea about some of this stuff than I did. So I think scientists do have a sort of tendency to oversimplify things to the general public. And something that was pointed out recently to me is that there is sort of no such thing as the general public. We often talk about like, I'm doing this presentation, I need to make it sort of accessible to everyone. But the people in the crowd aren't there because they have no understanding of marine science, they have no interest in marine science. They're there because they are interested. And we, sort of tend to oversimplify things, I think, and don't give enough credit to the general public where the credit's due, that they actually do have a lot better idea of marine science than sort of simplifying it to the point that it's inaccurate can be, sort of a bit of a problem.
Well, I hope with this podcast we push it a little bit further than just layman's terms. Do you think scientists should be trained in media and communication skills?
Yeah, definitely. What's the point in doing science if we're not sort of communicating it with policymakers and with the general public? Like, a lot of my funding is through government departments, so it's taxpayer funded research. So I think part of the trade-off of that is the taxpayers need to know what we're spending their money on. They need to be able to sort of see the value in the research that we're doing. And you can only do that if you're communicating effectively and efficiently. So I think, definitely need better training for a lot of marine scientists. Well, a lot of scientists in general.
Do you think the world has changed with, how much we know about sciences since social media and podcasts and the accessibility of information?
I think it definitely is. The number of times I'll just be sort of scrolling on apps at home and all of a sudden there's a video from a scientist that I know working on something in Melbourne or this and that. So I think we are definitely getting better with communication, and things like sort of short videos, podcasts, radio, making it a lot more accessible, and it sort of breaks down the sort of stereotype of the ivory tower of academia where, everyone's walking around in their sort of big posh gowns or like Harry Potter. Science isn't like that nowadays. It's definitely a lot more accessible, and people in science are realizing the importance of it too. And sort of there's a lot more work being done into that science communication field, which is, really great. Yeah,
it's not just a thesis put on a shelf and nobody gets to access it anymore. You can put it down to bite-sized chunks and get it on socials. So socials aren't always the devil. As much as I think they are.
Yeah.
And that's our social clip for this episode. All right, Morgan, if you had unlimited resources, what would your dream project be?
Oh, that's a challenging one. I reckon it would be coming up with just some better method of eradicating marine species. It hasn't been done before. Surely if we throw enough money, we can sort of start to get rid of invasive species as they arrive, similar to what we can in the terrestrial environment.
So where can people find out more about your work?
I suppose, through some of the social media channels like LinkedIn and stuff. And if people are interested in invasive species in general, the federal government has a marine pest website, as does most of the state governments. So if you see something that's not supposed to be there, you can report it through these websites and send it through to them. And they would rather people send through photos of species that are meant to be here rather than we miss the ones that aren't meant to be here. So there's a lot of divers out in the water. There's a lot of people with, GoPros up to big camera rigs that are really valuable in this biosecurity space. And the other sort of big area that can help is through things like iNaturalist, where you can upload photos of marine species and then people all around the world can help ID them. And when something gets detected that's not meant to be there, it alerts sort of the management authorities and the government departments that this thing's all of a sudden here, and sort of they can get onto it. So even if you're not certain about your ID, just upload it to iNaturalist and you never know what might happen from it.
So what's that website?
That's iNaturalist.org. So it's great for, people all around the world can ID whatever species you put up, native or otherwise. And it helps biosecurity researchers. It alerts government authorities. It can also help sort of just broader research. You can get sort of a research grade photo out of it, and then we can use that to identify where native species are, where they're moving. So it's a really great resource for citizen scientists.
Great. Well, Dr. Morgan Ellis, thank you very much for coming on to Ascend Your Story.
Thank you for having me.
Thanks to the Ascent Media team for putting this show together. Producer Jonathan Brown, all things production Michael Nguyen, and intro sequence by Nico Scott. I'm Sam O'Reilly. Ascend Your Story is an Ascent Media production. We tell the stories of amazing people in science, tech, research and more through video, photography, live streaming and podcasts like this one. You can see our work and check out all the other episodes at ascentmedia.com.au.